Hello my dear students, in this video we are going to understand about the transportation of lipids along with the metabolism topics are covered. Please watch till the end of the video so that you can understand each and everything. If you are new to my channel, please subscribe and share this with your friends. So for the lipid transportation, we have to understand first how the lipids are soluble in the blood. The lipids are the compounds which are insoluble in the water. That means the lipids are completely hydrophobic. They do not soluble in the water. So whenever they are not soluble in the water, how can they soluble in the blood? So this is a problem, right? The blood containing the aqueous environment in which the lipids are not at all soluble. How this problem can be solved? So we have the physiological solution for this problem. That means lipids are binding to the apolipoproteins. So after the binding of the apolipoproteins, they will form the compound called as the lipoproteins. Lipids are binded with the proteins. Lipids plus proteins will form the apolipoproteins. These apolipoproteins are helping to bind with the lipids, finally leading to the formation of the lipoproteins. So these lipoproteins containing the specific components in it. One is hydrophilic shell. It is soluble in the water. One more is the hydrophobic core, which is present in the inside region where it is not soluble in the water. You can see this green color structures, right? It is a phospholipid. And this yellow color structures are the free glycerols. And in between there are triglycerides. That means they have the glycerols and fatty acids. And also you can see the cholesterol esters. That means cholesterol is binding to the fatty acids, leading to the formation of cholesterol esters in between of the structure. And you have the main apolipoprotein. That means these proteins are bonded with the triglycerides and some of the lipid components, leading to the formation of the lipoproteins. This complete structure is helpful for the transportation of lipids in the blood. We have to understand that there are the fundamentals in the lipid transportation. Here, first, we already know that the lipids are transported from the intestine to the liver or into the targeted organs. Whenever we see, depending upon the protein content, the lipoproteins are different types. So I will show you. If the protein is high, then it is high density lipoprotein. If the proteins are low, then it is low density lipoprotein. This is the picture. You can see that here. This is HDL, high density lipoprotein. LDL involve low density lipoprotein. MDL is mediumly dense lipoprotein. VLDL is very low density lipoprotein. And chylomicrons, where we have the temporary formation of these structure of chylomicrons, is formed after the digestion of lipids. Okay. First we have to understand what is HDL. HDL contain more amount of lipoproteins. So it is called as high density lipoprotein. Here you can see the red structures. The red structures are the apolipoproteins. These are present in the greater number. So it is called as HDL. In the LDL, we can see there is higher amount of the cholesterol. That means the proteins are less but the lipids are more. In the medium density lipoproteins, the cholesterol is in the medium amount. First you see the cholesterol in the HDL. HDL, the cholesterol levels only 15%. But here in the LDL, the cholesterol level is higher. And in the MDL, that is medium density lipoprotein, the cholesterol is not more, not less. It is in the medium level. But in the very low density lipoprotein, there is higher of the triglycerides, cholesterol esters, total free cholesterol. Everything is in the higher components. And next, in the chylomicrons, we have the same similar amount of the triglycerols, cholesterol esters, and free cholesterol is very less, but phospholipoproteins and apolipoproteins are greater. That means from the chylomicron, these chylomicrons will convert into the VLDL. See, this is just an overview. I will explain you in detail. So you can see the structure of the low density lipoprotein. Here this is an apolipoprotein. That means this is a protein which is attached to the lipids. You can see the cholesterol esters. The cholesterol is attached to the, here this one is the cholesterol. It is attached to the free fatty acids, and you can see the free cholesterol, only cholesterol is there. And in the inside you can see this triglycerides. Okay, this one is glycerol, this one is the fatty acids. You can see it, right? So this is the simple structure of LDL. In the HDL you have the 3 lipoproteins, right? You can see in the HDL more amount of protein is present when compared to the lipids. So you can see the cholesterol here, the cholesterol here, this is the cholesterol again. Here you can see the triglycerides, and this yellow color structure is phospholipid membranes, phospholipid layers. So this is the overview of the classification of the plasma lipoproteins. First we will understand from where they are formed and what is the main role. The chylomicrons are formed from the dietary triglycerols, and these chylomicrons are transported from the dietary lipids to the tissues. I will show you the picture, then you can easily understand. So you can see that from the food these fats are entering into the intestine. After the digestion process, these will contain the lipids and some of the components of the lipids. All the lipids and components of the lipids will assemble together for the formation of chylomicrons, right? From the intestine, the lipids are assembled in the form of chylomicrons, and from the intestine, these chylomicrons are entering into the bloodstream. Okay, so this is the fate, that is the role of the chylomicrons. We will understand VLDL. Very low density lipoproteins. It is produced from the endogenous triglycerides. That means our body contains the fats which is present in the liver. From the liver, these triglycerides are transforming into the VLDL. From there it will reach the tissue. I will show you the picture. Here you can see the liver containing the VLDL. This VLDL is transported into the bloodstream. From there the VLDL enters into the capillaries, and there after the enzymatic action of the lipase, the VLDL is broken down by the enzyme called as lipase. From there the VLDL will, after breaking down, it will produce the free fatty acids. These free fatty acids will enter into the muscles or enters into the mammillary glands and adipose tissues. These all tissues will store the fatty acids. So here liver release VLDL, VLDL enter into bloodstream. From the bloodstream it will break down by the enzyme lipase. After the breaking down it will release the free fatty acid. These free fatty acid are going to the adipose tissue. From the blood it is reaching adipose tissue, muscles and mammillary glands for the storage. Okay, so here this is the fate of the VLDL. Again, after the breaking down of the VLDL, some of the remnants are released. Okay, so here the chylomicrons are also broken down. Some of the remnants, that means the leftovers. After the breaking down of the VLDL and the chylomicrons, the leftovers are released. These leftovers are reaching back to the liver. You can see here the chylomicrons reaching back to the liver, and VLDL is also reaching back to the liver. Or these remnants, these are the leftovers, they will convert into the LDL. Okay, this LDL is again transported to the extrahepatic tissues. That means some of the tissues include the adipose tissues, muscles, etc. These are also leading to the storage of the fats. Okay, I hope you understand. The remnants of the chylomicron and the remaining products of the VLDL, either they will reach to the liver or this VLDL is converted into the LDL. From the LDL it is stored into the some of the tissues, or again this LDL is reaching to the liver. So this is the fate of the VLDL and how it is being metabolized in the body. I hope you have understand it clearly. Next one we will see about the LDL. So this is the low density lipoprotein. Here the cholesterols are converted into these lipoproteins, low density lipoproteins. This delivers the cholesterol to the peripheral tissue. From the body, it is been stored into the peripheral tissue. That means we have the belly fat, we have the fat in the thighs, or we have the fat in the back region. So this all fat is been stored by the cholesterol only. This cholesterol is converted into the peripheral tissue bad fat. So this is also leading to the excessive increase of the body fat storage. So this LDL, I have said you know, the LDL, okay, I have said you right, the LDL which is produced from the VLDL remnants, or the LDL which is containing more amount of cholesterol from the food. From the food, the cholesterols are released, and these released cholesterols leading to the formation of LDL, or the VLDL is converted into the LDL. That is going into the adipose tissues or extrahepatic tissues for the storage of excessive fat in the body, or this LDL can reach back into the liver for the metabolism. And next, we have to understand about the HDL, high density lipoproteins. This HDL is formed by the most of the proteins and phospholipids. Here you can see there is more amount of proteins and more amount of the phospholipids. This is an HDL, right? What happens? What is the function of HDL? When our body is containing more amount of HDL, this will reverse back the cholesterol back into the liver. That means, I will explain you. Our adipose tissues is having more amount of fat. I have said you, the cholesterol is converted into the fat in the extrahepatic tissues. The HDL precursors are released from our liver. Our liver and our intestine will produce more amount of HDL, and also the precursors of HDL which are converting into the HDL. What is the function of the HDL? This will take the cholesterol back into the liver. That means whatever the useless cholesterol, the bad cholesterol which is present in the body, it is taken back into the liver. In the liver, the cholesterol is broken down and it is used for the further product development. That means HDL is good cholesterol and LDL is bad cholesterol. If we have more amount of LDL, the bad cholesterol is stored in the body. If we have the more amount of HDL, the good cholesterol is stored and the bad cholesterol is removed with the help of the HDL back into the liver. So this is the general overview of the different type of the lipoproteins and the metabolism transportation of the lipids. This is overview. Reading this much is more than enough because they will not ask in depth in your entrance exams. So just you have to know what is chylomicron, what is VLDL, what is LDL, what is IDL, intermediate lipoprotein. Next, what is HDL? So this everything you have to understand how these are transported and which one is good, which one is bad, which one containing the more amount of proteins, which one is containing more amount of cholesterol. So you have to just remember as an overview only. So if we see the chylomicrons, you just remember the chylomicrons is mainly composed of the fats, right? So these free fatty acids, triglycerides, free glycerols, glycerol esters, these are present in the chylomicrons. The chylomicrons is formed in the body. You can see if we take the blood test, in the plasma we can see that after the consumption of the fat, the blood will turn into the milky appearance after 1 hour. Okay, so after 1 hour it will turn into the milky appearance because of the presence of the chylomicrons in the blood. So this is the fate of the chylomicrons. We take the food containing the fat. The food enter into the intestine. From the intestine, the chylomicrons are acting. These are broken down by the these lipases enzyme. After that, they will release the triglycerides. The remaining products of the chylomicrons, which are remnants, they will enter into the liver. So this is the general fate, general metabolism of the chylomicrons. VLDL. We already know that in the very low density lipoprotein, we have more amount of triglycerides, cholesterol esters, free cholesterols. So these are the more amount of the fatty acids. Okay, predominantly the triglycerides are present in the more amount of content. So they are called as VLDL. So VLDL is synthesized by the liver, and this liver releases the VLDL. You already know that the chylomicrons, after the food, they will enter into the bloodstream. The liver will produce the VLDL. The VLDL and the chylomicrons are broken down by the enzymes. There will be a remaining components. They again reach back to the liver. Again, the VLDL remaining components, they will reach back to the liver, or they will transform into the LDL, and after the breakdown we will get the final product called as free fatty acids. After the breakdown of chylomicrons and VLDL, we will get the free fatty acids as a final product. After the free fatty acid formation in the blood, they are stored in the muscles, adipose tissues or mammillary glands. This is the general explanation. The lipoprotein breakdown, how the lipoproteins are broken down in the tissue. I already said you, our body having the muscles and adipose tissues, the fat is stored in these tissues. How the fat is broken down? The fat is broken down by the enzyme called as lipoprotein lipase. This enzyme will break the fats. Okay, these fats will release the fatty acids into the blood circulation. So after the broken down of the fats, these triglycerides are broken, right? They will release the chylomicrons and VLDL into the blood circulation. Whenever the free fatty acids are formed finally, they will produce the energy, right? Whenever there is need of the energy, when we don't have glucose, then the fats are broken down. When the fats are broken down, they will, the storage fats, the triglycerides are broken down into the free fatty acids. These free fatty acids finally will release the energy to the body. We already know that the triglycerides contain the glycerols and also the free fatty acids. Then what happened to these glycerols? The free fatty acids will produce energy, but what happens to the glycerols? These glycerols are not able to metabolize in the adipose tissue. So these glycerols will enter the blood. They will enter the blood. From the blood, the glycerols will enter into the liver. So this is the fate of the glycerols. Next, we will understand the metabolical pathway of the lipids. That means we have to know overview of the metabolism of the lipids. First, we already know that the lipids are absorbed in the small intestine. They are transported with the help of chylomicrons. Chylomicron transport these triglycerides from the intestine, and there they will undergo the different type of metabolical actions. We already know that VLDL is very low density lipoprotein, which is containing more amount of the lipid complexes. This VLDL is helpful for the transportation of the more amount of lipids called as triglycerols. The VLDL is released from the liver. From the liver, the VLDL is released into the blood, and from there it will reach to the different type of body parts for the better storage. Adipose tissue. We already know that we have the fat in the body. The fat is stored inside the adipose tissue containing the adipocytes. These are the cells which contain more amount of the fats. Okay, so these fats are stored in the concentrated form in the adipose tissue. They act as an energy reserves. Whenever there is a need of energy, these fats are broken and they will release the energy. So triglycerides are broken down to release the energy from the adipose tissue. Utilization of energy. How the energy is finally produced. Whenever there is a breaking down of the fats, the final end product is fatty acids. That means the tissues will produce the final fatty acids. From there, the energy is released. So this will help our metabolical demands to complete the functioning of the organs like hearts, muscle, etc. When there is no glucose, the fat which is present in the body will break to produce the energy, because our body continuously work with the help of the energy called as ATP. So how the ATP is generated, I will show you the picture. So whenever our body is containing the stored triglycerides, these triglycerides are converted into the VLDL. This VLDL enters into the blood. Okay, or else whenever the triglycerides are formed, they can be re-esterified into the liver for the formation of the large chain fatty acids. That means this is helpful for storage. This step, re-esterification, is helpful for storage, but the breaking down is helpful for the release of the energy. Don't go in depth. This is an easy concept, you can able to understand in easy way. So continuously, if there is a breakdown, then we will have the continuous supply of the triglycerols. The triglycerols which are stored in the adipose tissues, they break down for the formation of the free fatty acids. These free fatty acids will help to produce the energy for the skeletal muscles and the cardiac muscles, because they are continuously working in the body. Approximately 65% of the free fatty acids are not oxidized. When the free fatty acids are not oxidized, what happens? They are re-esterified. They are coming back. Again, they are building back into the large chain fatty acids. So, the amount of the fatty acids which are used up. The remaining will go back into the re-esterification. Again, they will form the large chain fatty acids. Again they are stored, okay, in the body. This is a continuous cyclic process of the triglycerides to release the energy. There are the four main fates of the intercellular lipids. Whenever there is a fatty acid or monoglyceride is formed in the body, then what is the fate of these lipids? After the formation, after the breakdown of the lipids, then what happens? What is the fate? We have to understand. Immediately after breaking down of the lipids, they will produce energy. When there is no use of energy, they will store. Okay. When there is a need of energy, they use it. When there is no need of energy, the remaining energy is stored. And after the storage, some of the body parts are made up of the lipids, right? Like cell membranes. The cell membrane which is present around the each and every cell is made up of lipids. Or these lipids are also helpful for production of the cholesterol. So you have to see that when the lipids are broken down, they use as fuels in the body, or whenever the lipids are more in the body, they are stored in the adipose tissues, okay, or they will form the cellular membranes, okay, phospholipid membrane, which is helpful for protection and integrity of the cells in our body. See, you have to understand that cholesterol is made up of the lipids only. The liver is producing the cholesterol by using up the lipids. Next one, fatty acid oxidation. So we have to understand the fatty acid oxidation. In the carbohydrates, we have the glycolysis, glyconeogenesis, gluconeogenesis, etc. Here we have to understand in the lipid metabolism, there is fatty acid oxidation process. How the fatty acids combining with the oxygen, they will produce the carbon dioxide, water and ATP. That means for the production of energy, the fatty acids are to be oxidized in the presence of oxygen. The fatty acid combined with the oxygen, they will produce the CO2 as an end product along with the water, and final necessary ATP energy is generated. So we have to understand what is beta oxidation. The beta oxidation of the fatty acids. Here, the breakdown of the fatty acids is called as beta oxidation. Why we are telling as beta? Because whenever the fatty acids are broken down, they break between the beta and gamma carbons. Okay, so that is the reason, because they are breaking at beta and gamma region, it is called as the beta oxidation. So what happens in the cleavage process? this is a continuous oxidation. It is a repetitive breakdown of the 2 carbon units leading to the formation of the free fatty acids. That means the shorter fatty acids are produced. After the production of short fatty acids, these short fatty acids converted into the acetyl-CoA. So after the formation of acetyl-CoA, this acetyl-CoA will enter into the TCA cycle, where after the TCA cycle, the acetyl-CoA is breaking into the different type of compounds, and finally it enters into the electron transport chain. After the electron transport chain, the final ATP energy is formed. So what are the steps in the beta oxidation? Activation of fatty acids. The fatty acids are activated. Okay, so in the cytoplasm, the fatty acids are activated. Okay, and there is an enzyme called as acetyl-CoA synthase. This enzyme is helpful for activation of the fatty acids for the formation of fatty acyl-CoA. Here we have the fatty acyl-CoA, where the long chain fatty acids are to be broken down. They enter into the mitochondria. From the cytoplasm, the activated fatty acid enter into the mitochondria. From the mitochondria, they undergo the beta oxidation cycle. That means the activated fatty acids are continuously broken down by the different type of reactions called as dehydrogenation, hydration, oxidation, thiolysis. Everything occurs in this beta oxidation cycle, where the free fatty acids are finally forming the acetyl-CoA. This acetyl-CoA enter into the citric acid cycle. It is also called as TCA cycle, where finally this produce FADH2 and NADH. The acetyl-CoA converted into the FADH and NADH, these enters into the electron transport chain ETC cycle. From there, these are used to produce the ATP. So finally, we are breaking the fats to produce the energy only. So final end product is energy only. That means in the form of ATP. So this is the overview of the beta oxidation. This is the definition. The primary end product. Okay, the fats are produced finally into the acetyl-CoA. So beta oxidation, final end product is acetyl-CoA. The fatty acids are broken down into the 2 fragments, and these are again broken down into the acetyl-CoA. So this acetyl-CoA is a 2 carbon fragment which is again entering into the TCA cycle. We have to know that there are 3 stages, right? Activation, transportation, oxidation. These fatty acids are activated. After the activation, in the cytosol, they will enter into the mitochondria. This is called as transportation. From the cytosol to the mitochondria, they are being transported, right? So it is called as the transportation. Next, here the beta oxidation. These activated fatty acids are oxidated in the mitochondrial matrix, finally to produce the energy. So this energy is used in the skeletal muscles, heart or the liver. Wherever the energy is needed, there the fatty acids can be used. So what happens to the final acetyl-CoA? I already said you, the final acetyl-CoA leading to the citric acid cycle or Krebs cycle or the TCA cycle. The final acetyl-CoA is formed. Right, after the fatty acid oxidation, the fatty acids are breaking into the acetyl-CoA. This enters into the TCA cycle, where the TCA cycle will produce the final CO2, H2O and adenosine triphosphate. After the TCA cycle, this ATP is generated. So this is a fate of the acetyl-CoA. This happens to the acetyl-CoA after the breakdown of the fats. So this acetyl-CoA can also help in synthesis of the cholesterol. You have to remember the point. So this is the general overview. This is the basic knowledge you have to understand. Here the carbohydrates will produce glucose, right? The glucose is breaking in the glycolysis pathway, right? The final product is pyruvate. So this is a metabolical process of carbohydrates. The fats are broken down into the fatty acids. Here in the fatty acids are broken into the acetyl-CoA. That means here undergoes the beta oxidation. After the beta oxidation, the fatty acids converted into the acetyl-CoA, and here the proteins will break down into the amino acid. From the amino acid, they will enter into the acetyl-CoA forms, and then the TCA cycle. The acetyl-CoA is the key product from the glycolysis, from the beta oxidation, from the transcription processes. This acetyl-CoA is formed. This acetyl-CoA is entering into the TCA cycle, where these products are formed, leading to the final electron transport chain process. In this electron transport chain process, finally the ATP is being generated. You can see, right? So finally acetyl-CoA go to the TCA cycle. From there it will go to the ETC cycle. From there the ATP is generated. So this is the main explanation. I think if you have understand, please support me. Thank you, my dear students.